In this presentation, I would like to share about screening of somoclonal variation in banana Musa acuminata through RAPD marker system. This is taken from a publication from Muslim Razani et al. The research trying to figure out the type of morphology on the tissue culture when they use different concentration of BAP, when they increase the concentration of BAP. So the normal control here is for 0. You can see that after the 22 subculture, the percentage of scalp actually finally resolve and grow into a normal shoot. And by the 25th subculture, all of the scalp structure initially develop into the normal shoot for 0% for no BAP for the control. But with 5, 10, and 15 milligram per liter of BAP, we can see that the number of normal shoot is 0. But only 5 and 10 actually develop into a rosette-like structure. Whereas for the one that is the highest concentration of BAP, it never developed into the normal shoot nor as the rosette-like structure. So as you can see in this picture, it shows the visual presentation of what happening in the tissue culture of banana when you increase the concentration of BAP. So with control with no addition of BAP, after the 25th subculture, it developed into the normal shoot. But then for 5 and 10, only after the 24th subculture, most of them actually develop into the rosette-like structure, where the leaf actually have the formation of the leaf. But then for the one that is highest concentration of BAP, 15 milligram per liter, we can see that it never developed into rosette or the normal shoot. That means there is no leaf and it remains in a scalp structure. So the research question that we want to find from this is that we can see that from the clone there is changes morphologically when we treat it with the different BAP concentration. So the question here is there any possible genetic change that follows with the phenotype that we see? So we want to find whether there is or not a genetic variation. So the keyword here is the genetic variation between the clones, because initially all those clones that were used in the treatment were initially from one mother clone. So genetically they are supposed to be similar. However, when we treat with different BAP concentration, it shows different morphology. It shows different phenotype. So the question here is there any genetic change that follows the morphology changes that we see? So we want to detect the variation at the genetic level using DNA marker. So the DNA marker of choice that we choose for the study is RAPD, which is stand for Random Amplified Polymorphic DNA. So just to recap here and just restate the research objective. For this experiment, the research objective is to investigate the possible genetic change among the banana tissue culture developed using different treatments of BAP at the 25th subculture using RAPD DNA marker. Before we move even further into the experiment, I just want to give you a brief introduction about RAPD marker system. So RAPD marker system is just what we call PCR amplification based DNA system, where they actually use a PCR method, which is polymerase chain reaction method, in order to amplify certain part of the DNA by using random primer. So what is a primer? Primers are those uh, what we call prefix and suffix inside the DNA that we use in order to tag a specific region within the DNA that we want to amplify. So whenever it has a pairing to the DNA, to the genetic background, then it will pair, and then the amplification will start to happen when it has both reverse and forward primers at that specific location. when it is not too far apart. If it bind into somewhere that is too far, then it will not forming any amplification too. It has to be, the size has to be just nice enough for amplification. So with RAPD marker system, they actually use a random primer in order to amplify certain region of the genetic that can give indication to us to the genetic variation that may have happened, and we can use this information in order to know the variation between individuals. So we can answer the research questions that we have initially with our banana clues. So as you can see here, with RAPD marker, so the blue one actually is the marker. So it actually tag the total genomic DNA of the plant. It tag a specific region, and if the amplification happen, it will actually produce a band that can be separated in agarose gel. All right. So based on the DNA, so this is actually the line, the red line and the green line right here that you see in this yellow box is actually the DNA that has been separated in agarose gel. Okay, we'll show you later on. So it will actually produce band. When we do this analysis, it will produce a band, and different genetic background can give different possible variation to the type of bending that appear and may not appear. All right. So if the background are all the same, they're supposed to produce the same type of bending marker. So maksudnya when it has band here, it's supposed to have band here also. If it has a band here, then it has supposed to have a band here. So maksudnya the bending pattern here and here is supposed to be the same. But then if the genetic background already changed, there are certain part of the primer that cannot be amplified, so it will not produce any band. It will not produce any amplification. It will not amplify that specific region. So when it's not amplified, it will not produce any band. But then that primer bind to another type of to another location of the genetic, then it may produce a different type of bending pattern. That means it produces a new amplification product that were not initially amplified before in the first organism that we use as a reference. So that reference can be the mother plant. Alright, initially before treatment. So it becomes as our control. So in this experiment, what is required? The first one that is required is the callus itself, the sample itself. So the sample that has been treated at the 25th subculture. So the example here is at the 20th subculture, but the one that is used for the early extraction is actually the one at the 25th subculture. So I just show you the picture example here. So we want to use the callus at the 25th subculture from all the treatment, from the control, from the 5, from the 10, and from the 15 BAP. And we actually extract DNA from the callus. So the extraction of DNA will actually carry out using a plant DNA extraction kit from Electrophoresis. Alright, so we use the extraction kit uh, in order to extract the total genomic DNA from the callus. Alright. Other thing that we need is a PCR amplification kit. So what is in the PCR amplification kit? We'll go over that component later on. So there are several components in a PCR reaction kit that is required for amplification to happen. Another thing that we need is actually the primer, because we are using our RAPD marker system. With our RAPD marker system, there are several set of primer that you use in order to generate that bending pattern in order for us to differentiate and identify whether or not there are genetic variation between the clones. Alright. So the tools that we use in order to see the DNA is the agarose gel electrophoresis. So the thing how this thing works, this is actually a gel, agarose gel. So the blue one is the agarose gel. So as the name actually imply, agarose is actually like the agar, macam like the agar-agar that we eat. Alright, so it has like a jelly-like structures that interlink with each other. So it will actually has a well inside here, a lubang, like a pari. You know, that's a well. So sample well, that 4 right there that you see. So that's where we load our sample. So inside here, it will actually be this gel is actually immersed inside a solution. It immersed inside the solution that react as a medium for the electrolyte, for the electric supply to actually move from one direction to another. So this electrode is actually negative charges at this one, at this side, and at this the other side is a positive charge. So the electrode will move from negative to positive. Alright, so that actually is important, that the that the charges of the DNA is put on negative side. Alright, so that it also moves towards positive electrolyte. So the direction of movement for DNA is at this direction, like the arrow show right there. So when you actually put the DNA inside the sample well here, and you run the power supply, the ion will move from negative to positive, so as the DNA inside the well. The DNA inside the well will also follows the ion, so the electric charges from negative to positive. So the DNA inside this gel will also move from this to this. How it move? It actually move following dia punya size, following the size. The bigger the DNA is, the slower it actually moves inside the agarose. So those of the small size DNA will actually move faster, just like human, just like all other stuff, following the gravity. So the lighter you are, the faster you move. Right, so it's the same as the principle here with the DNA. So the small DNA inside the well, when we load them, they're going to be have multiple variations of size of DNA depending on the source of DNA that we run lah. If it's a PCR product of RAPD just now that we're talking about, they probably have few bands. Alright, so that band will actually separate with each other, and the separation is actually based on the size. So the smaller one will actually move faster than the bigger one. So basically, the experimental setup for the whole experiment is just as summarized in this chart right here. So we first going to start with DNA extraction of the callus, all right, from all those treatments 0, 5, 10, and 15 at the 25th cell culture, and then we're gonna do the DNA extraction using the kit, the plant DNA extraction kit. And then from the DNA that we extracted, we're going to use that DNA in order to run gel electrophoresis, as I explained later on. This is in order for us to see how intact is our plant genomic DNA. And then we're going to run from the total genomic DNA that we extracted. We're going to take some in order to run gel electrophoresis. And then we're going to take another sum from this in order to run PCR using that primer, the RAPD random RAPD primers. We're going to use the primers in order to amplify region and to produce bending pattern. Alright, so using the technique of PCR, polymerase chain reaction, we're going to go over the technique after a while. And then we have the PCR product. The PCR product will run again through gel electrophoresis in order to see the bending pattern. Before anything, before we can actually get the DNA for the analysis, we have to extract the DNA from the plant sample. So in the sample for this analysis is going to be the callus or the tissue culture sample for the banana. So we're going to take a bit like 10 gram of the callus sample, and we're going to grind them into powder form. Alright, so depending on the method of the mechanical method that we use in order to grind our sample, it can either be in a dry or wet form. So if we use liquid nitrogen, it usually will turn into a dry powdery form, but within a minute it will turn wet after the liquid nitrogen has dissipated. Alright, so we grind them into powder form, or if we use ethanol, it's gonna be a small break plant sample in ethanol in a wet form lah. And then we're gonna add a lysis buffer. So the lysis buffer here function in order to break all the components of the plant cell and release all the material from the cell out. Alright, and then we're gonna add solution in order to remove protein, separate protein from the nucleic acid. And then we're gonna add solution in order to precipitate the DNA. So this most likely will actually include solution like ethanol in order to separate the DNA from the rest of other molecules within the cell. And then we're gonna rehydrate, we're gonna remove all the solution and remain only the DNA inside the tube, and it will be rehydrated in whatever buffer of choice lah. So usually this actually can be either TE buffer or water. So in order to make things simplified, we're just gonna say that the genomic DNA solution can be done using a kit. Alright, so in this case, we're gonna be using GeneJet plant genomic DNA purification kit. So for DNA extraction, it can either be two-way lah. It's either you using a plant DNA extraction kit, which is a commercial kit that can be bought ready and you just mix according to the instruction, or you can actually prepare the buffer, the cell lysis buffer, the protein removal buffer, or the DNA precipitation buffer yourself. Alright, but in this case we are using kit. So in this picture here, it actually show the type of DNA, the how the DNA will look like after extraction. So in picture A, this is actually the genomic DNA when it is in the precipitated solution. When we precipitate them using an organic solution like the PVP, ethanol, or isopropanol, all these solution can actually be used in order to precipitate DNA from the solution. So we're just going to add equal amount of organic solution to the aqueous solution where the DNA is located in lah. So after that, once we see this strand of DNA like this, so that means we successfully extract the DNA from our sample. So we just can remove all the solution and it will remain as a white substance like in this tube right here. So we're gonna centrifuge this down using a centrifugation method. We're gonna centrifuge it down, and then it will actually appear as a white solute like this. So just if you're not done your extraction well, your DNA might turn yellowish like this, especially when the sample contains a lot of phenolic content. Alright, but this is A and B is a normal picture for DNA extraction. So this is what you expect to see lah. So just now the white strand that you see in the tube is what is visible to your eye. And then we want to see whether the DNA that we extracted is intact or not. Then we can see from this picture right here when we run our gel electrophoresis. So this is a gel electrophoresis. What gel electrophoresis is just it has a tank, alright, where you put the buffer inside and you immerse a gel with a well inside the solution. Alright, so it will have an electrode, a negative and positive side. So the DNA will be on a negative side. So when you turn on the power supply, the electric will run from negative to positive. And at the same time, when it runs from negative to positive, the movement of electric is going to be this way. This is going to be the direction of the electric. So it's going to pull together the DNA inside the well. So you're going to put your DNA sample that you extracted just now inside this well. Alright, so when you turn on, it will pull together the DNA. So what will happen is what is pictured on the left right here, on the right here. So when you have a DNA of a different size, it will turn out like a ladder on the first well right here. As for your extracted genomic DNA, because it's just one DNA extracted from your sample, you just gonna see one band, single band for each of the sample, and it's gonna be at the top compared to the ladder because it is of a big size. So most of the, it says gDNA here is because it's genomic DNA. So most of the genomic DNA, if you did not do anything and you done your extraction well, there is no shearing, there is no cutting of genomic DNA, then it will appear intact, one band like what we see right here. So this is just our control that we use as reference in order to know the size of the genomic DNA. Alright, so it has to be at the top, even bigger than our reference, because genomic DNA is supposed to be large. Alright. So after you have made sure that your genomic DNA is all intact and fine, then you can actually use that as your DNA sample in a polymerase chain reaction. What is a polymerase chain reaction? Polymerase chain reaction is actually an amplification method of a specific site of DNA. Alright, so what is required in a PCR reaction? You need your DNA sample. Your DNA sample here is going to be your DNA that you extracted from your banana callus. Alright, and then you need a primer. What is a primer? A primer is a 9 mer, it means 9 nucleotide of DNA on a forward and a reverse that gonna specify where you want to amplify a specific region within your genomic DNA. So if you have your sample here, your genomic DNA, then the primer will actually bind where it complement to each other, and then it will actually specify a certain area within the genomic DNA that you want to amplify. So it requires a primer, and then it requires a nucleotide as a basis that will actually to form that amplification. So those nucleotide is the nucleotide that require in order to make the DNA lah, that will be amplified. So it's ACGT. Alright, and then you require a PCR tubes, a mixed buffer. So a buffer is just to recondition the suitable condition for amplification to occur, and then a tag polymerase. So if you recall in your basic science that you learned before this, polymerase, DNA polymerase is the one that actually do the amplification. Alright, so DNA amplification, replication within your body, then it's the same. It's the same polymerase. It's the same polymerase that synthesize DNA. Alright, so tag here is just to specify the origin of the polymerase that is used in order to make the PCR. So it's from the species tag. Alright, so tag polymerase is the DNA polymerase that's gonna perform the amplification. So in the PCR process, it's just a simple tree method that's going over and over again until you get numerous number of amplification of that specific site of DNA. So it's gonna start with denaturing, where it usually performed with 95 degrees Celsius in order to separate the strand. Because DNA is double bond, you want to separate them, so then the primers can go and bind to that specific location during the annealing punya time. So after 95 degrees Celsius, then the temperature will lower down to 55 degrees Celsius in order for primers to find its complement region and bind. And then it will increase the temperature again to 72 degrees Celsius, where the amplification will happen, the extension gonna happen. So this is the time when nucleotide will actually go in and find and complement, and the amplification will happen. So these three process will go over and over until the first cycle you're gonna get 2 copy of that template DNA, and in the second cycle you're gonna get um, 2 to the power of 3, 8 copies of that DNA template. And then in the third cycle you're gonna get 16 copy, and in the fourth cycle you're gonna get 32 copy. So the cycle will actually continue for about 50 times or 25 times, depending on your PCR programs lah. Alright, so this is where you actually get exponential amplification of that specific site within your DNA region, depending on the primer that is used in order to specify the location. So what primer are we using? Okay, so the primer that we'll be using is actually listed in this tab right here. So it will be, so this is the text that I actually extracted from the paper. So it will actually use this primer as listed in yellow and red letter. So as you can see here, it actually give all the component that were used in the PCR, and 14 different primers were actually being used in order to run the RAPD. One primer is for one reaction, so it will do in 14 different PCR reaction. So similarly, just now when we finish our DNA extraction, we also run gel electrophoresis. So after we run PCR, we also run our gel electrophoresis. And then in this picture right here for A, it's actually for primer OPA01. So if you can see, M is for leather. So this is the reference, as I taught to you previously, that's as a reference for us in order to know the size of our DNA band. Alright, so if you can see here, there is annotation there to specify the size of DNA at that specific region. So if you can see, the larger the DNA, the higher it is in terms of location, in terms of relative location to other DNA that are smaller, 100 pp. So 1, 2, 3, 4, 5, and 6 is just a different sample based on the table right here. So sample number 1 is normal shoot, and sample number 2 is the rosette-like structure, and then 6 is a scalp with the 15 BAP concentration. Alright, so for this one is for primer OPA01, B is for primer OPA02, C is OPA06, and so on. Alright, so one primer for one gel picture because it from one PCR reaction. Alright, so number 1, if you can see, it produced 2 band, all of it produced 2 band, with a number 6 has a slightly different size compared to the rest. Alright, so um this actually signify difference. Alright, so if you can see D for OPA15, you can see that all of the sample, it has band on the top. Alright, it's a bit vague, but you can see at the top there is band. But for sample number 1, which is control, there is no band at the top there. Alright, so that actually signifies variation. Okay, so same goes with primer for picture E, OPA19.
all of it has band at the bottom, but the control does not have band right here. So for the rest of other primers, it's pretty much the same with a slightly change right here. But in terms of presence and absence, they are all the same. So we can say that all the other primers are monomorphic and only D and E is polymorphic for normal compared to treated sample. Right. Alright, so same goes with primer OPC03 and OPH09. This one also show polymorphism with the absence of band for control. Alright, so the control compared to the treated samples are different. So this actually signified the DNA variation between the non-treated sample and the treated sample. So this is the text that I extract from the article. So as you can see here, it says that four primers out of 14 primers that were used in the amplification show a unique bending pattern that differentiate between the control and the BAP treated scope after 5 subculture. Alright, so the RAPD polymorphic band from the 4 primer actually signified there are a certain type of genetic variation that occur with the treatment, with the BAP treatments that they apply on the tissue culture. So in this lab report, students are actually required to write a simplified lab report based on the experiment. So these are among of the element that is required. So just a simple introduction to the experiment and the objective of the experiment, and then simplified methodology as what I explained. So you just put based on what I explained, and you can Google search a bit in order to add more to it. And then result and discussion. So I already discussed a bit on the result, but I will put up link on to the article so you can go over it, and then you have to discuss a bit its result. What is the significance of the variation that we see? So, and then after that, you can end the report with the conclusion of not more than 80 words, right? And then reference to what you have added to your lab report. With that, thank you for your attention.